Hello and a warm welcome to everyone to today's webinar on advanced strategies in bioanalysis of biosimilars achieving through robust assays. My name is Bhavika Maheshwari and I represent Veeda Group. Veeda Group is a full service contract research organization offering comprehensive end to end solutions ranging from preclinical to clinical and bioanalytical services to support innovator generic, and biosimilar drug development programs for our global clientele. We have significantly expanded our global presence across 26 geographies. Together, we serve clients globally in pharmaceutical and biopharmaceutical industries, agrochemical and industrial chemicals, herbals, nutraceuticals, and medical devices industries. As your facilitator for today, I'd like to introduce you all to a couple of pointers before we begin the presentation. First, we'll have the presentation by Dr. Jagdish, and with that will be followed by a Q&A round. We have enabled Ask a Question feature, which is right there on your screen. You can drop in all your questions there. Now, moving forward, I would like to actually introduce our panelist, Dr. Jagdish Bora. Dr. Jagdish Bora serves as a general manager and oversees the clinical bioanalysis department at Veeda Biopharma Division, which specializes in supporting biotherapeutics drug development. With a PhD in pharmaceutical sciences, Dr. Jagdish has over 17 years of experience as a scientific leader in regulated bioanalysis across various sectors, including pharmaceuticals, biotechnology, and vaccines. In his current capacity, Dr. Bora is spearheading the development and validation of methods for large molecules, as well as the analysis of samples using techniques such as chromatography and legal binding assays. Operating within a highly regulated and GCLP compliant laboratory, he is playing a pivotal role in ensuring the quality and accuracy of bioanalytical process. So, without any further delay, I would like to call Dr. Jagdish Boda to begin this session. Dr. Jagdish, over to you.
Thanks, Bhavika, for the introduction. Good evening, everyone. I'm truly excited to deliver today's webinar on advanced strategies in bioanalysis of biosimilars. So, over the next 40 to 50 minutes, we will be delving into the topic of bioanalytical considerations for the development of biosimilars. Next slide. So, coming to the agenda of this webinar, in the present webinar, I'm going to talk about, next slide, please. Yeah, and what introduction to the biosimilars. So, what is biosimilars and why biosimilars are important to the biopharmaceutical industry? And knowing about multiple biosimilar modalities which are currently available in the market, followed by requirements for approval of biosimilars. I also touch upon the clinical pharmacology studies to be conducted by utilizing bioanalytical strategies for pharmacokinetics, immunogenicity, and neutralizing antibody assays. And finally, critical takeaways or conclusions, along with some important regulatory guidelines to be considered for development of biosimilars, and then Q&A sessions. Next slide. So what is biosimilar? So a biosimilar is a biological product, generally large and complex molecules produced from living organisms, which are carefully monitored to ensure consistent quality. So by definition, as per FDA, the biological product is highly similar to the reference product, notwithstanding minor differences in clinically inactive components, and that there are no clinically meaningful differences between the biological product and the reference product in terms of the safety, purity, and potency of the product. So there are multiple definitions from FDA and EMA, but the overall meaning of biosimilars lies in that biosimilar has similar physical, chemical, and biological properties which are highly similar to the reference product. There may be minor differences from the reference product which are not clinically meaningful in terms of safety or efficacy. Minor variability is only allowed when scientific evidence shows that it does not affect the safety and efficacy of the biosimilar. So this will be achieved with a robust manufacturing process to ensure that all batches of the biosimilar product are of proven quality. Next slide. So why biosimilars? The biosimilars market is poised to grow over the next decade, and the size of the global biosimilar market is expected to grow from 25 billion in 2022 to about 126 billion by 2032. So, which is driven mainly by the increasing prevalence of cancer and the cost effectiveness of biosimilars. So, the use of biosimilars in cancer treatment has the potential to increase access to life-saving treatments for patients as they offer more affordable and accessible options for these critical therapies. So, as of March 2023, 49 biosimilars have been approved in the US, out of which 24 for use in cancer itself. Other indications also include infectious diseases, chronic and autoimmune diseases, blood disorders, growth hormonal deficiencies, etc. So there is a substantial increase in biosimilar startups and CROs, which are expected to play an important role in the development and commercialization of biosimilars in the coming years. So, as the demand for these treatments continues to grow and more patents on biologics are going to expire, so with the focus on innovation, cost effectiveness, and patient access, biosimilar startups and CROs have the potential to transform the biological biopharmaceutical industry and improve healthcare outcomes for patients around the world. So hence, there is a need to accelerate time to market, reduce R&D costs, and improve productivity for biopharmaceutical companies. Next slide. So coming to the different biosimilar modalities which are currently approved in the market, if you see the different modalities of biosimilars. So unlike small molecules, which are synthesized chemically and identical in structure and easy to characterize, most biological or biosimilar medicines in current technical use contain active substances which are made of proteins. These can differ in size and structural complexity from simple proteins like insulin or growth hormones to more complex ones such as coagulation factors or monoclonal antibodies. Sophisticated analytical methods are used to study the physicochemical properties and functional properties as well, so that molecular structure and protein modifications and biological activities can be assessed. So, due to the increasing complexity of therapeutic modalities for demonstrating biosimilarity, right from ADCs or bispecific or trispecifics and cell and gene therapy, so there is an increasing demand for developing more sophisticated analytical and bioanalytical assays. Next slide. So, a robust regulatory framework is designed for the requirements for approval of biosimilars by FDA and EMA. Usually, biosimilars are approved according to the same standards of pharmaceutical quality, safety, and efficacy that apply to all biological medicines approved in the US and EU. So, the aim of biosimilar development is to demonstrate biosimilarity. That means they should be highly similar in terms of structure, biological activity, efficacy, safety, and immunogenicity profile by conducting comparability studies. So usually comparability is conceived as a stepwise process that is tailored for each product. Knowledge from the initial quality comparability studies is used to determine the extent and type of non-clinical and clinical studies which are required in the next step of development, with the aim of ruling out differences in clinical performance between the biosimilar and the reference medicines. So if you see the comparative quality studies, these usually include comparing the protein structure and biological function using sensitive techniques capable of detecting minor differences with clinical relevance between the biosimilar and its reference medicine. These studies are more sensitive than clinical trials for detecting such differences, as there is often variability among human subjects participating in the clinical trials. So then once we prove comparability in quality studies, we have to perform non-clinical comparability studies, which include in vitro and in vivo pharmacodynamic studies, as well as in vivo toxicology studies. So these in vivo toxicology studies are only required in certain cases when the biosimilar is produced in a new type of cell or organism, or when the formulation includes new excipients which are not used previously. So once we prove comparability in non-clinical studies, we have to conduct comparative clinical studies. So clinical trials are tailored to confirm biosimilarity and to address any questions that may remain from previous analytical or functional studies. So a clinical study or studies that are sufficient to demonstrate safety, purity, and potency in one or more appropriate conditions for use, for which the reference product is licensed and intended to be used for which licensor is sought for the biological product. So overall, in evaluating a sponsor's demonstration of biosimilarity, FDA will consider the totality of the data and information submitted in the application, which includes structural and functional characterization, non-clinical evaluation, human PK/PD data, clinical immunogenicity data, and comparative clinical studies data. FDA intended to use a risk-based approach to evaluate all available data and information submitted in support of the biosimilarity of the proposed product. So in the subsequent slides, I will focus mainly on the clinical pharmacology study requirements and the respective bioanalytical assay strategies in generating comparative data for demonstrating biosimilarity with the reference compounds. Next slide, please. So one of the important studies for demonstrating biosimilarity or clinical pharmacology studies. These are built on the comparative analytical studies in the stepwise approach to demonstrate biosimilarity that there are no clinically meaningful differences between the proposed biosimilar product and the reference product. These studies may address residual uncertainties that remain after the analytical evaluation and can add to the totality of the evidence supporting a demonstration of the biosimilarity, and also guide both for the need and design of subsequent clinical trials to support demonstration of no clinically meaningful differences in the overall demonstration of biosimilarity. So what are the critical considerations in conducting clinical pharmacology studies? So a well-designed clinical PK/PD assessment is required to understand exposure-response relation to the biological products, which are very important for assessing whether there are any potential clinically meaningful differences between two products. So this evaluation should include assessments of PK similarity as well as PD similarity, which is applicable for the respective therapeutic agent. The selection of biomarker is very important, and it should be considered that the time of onset of the change in the PD biomarker relative to dosing and its return to baseline with discontinuation of dosing. Also, the dynamic range of PD biomarker over the exposure range to the biological product is important, along with the sensitivity of the PD biomarker and the relevance of the PD biomarker to the mechanism of action of the drug. A combination of PK and PD similarity can be an important assessment in demonstrating biosimilarity. Also, the need for additional studies at each step in this progressive manner will be determined by the degree of residual uncertainty that remains at each step regarding the similarity of the products and whether the study can address these uncertainties. Another important parameter is the extensive analytical characterization, which can reveal differences between the proposed biosimilar product and the reference product and see whether there is any insufficient analytical similarity or analytical similarity with residual uncertainty or tentative analytical similarity or fingerprint-like analytical similarity. So this fingerprint analytical similarity permit a very high level of confidence in the analytical similarity of the proposed biosimilar, and it will be appropriate for the sponsor to use a more targeted and selective approach to conduct animal and clinical studies to resolve any uncertainty and to support demonstration of biosimilarity. So when performing an evaluation of clinical pharmacology similarity, it is very critical to use the appropriate bioanalytical methods to evaluate the PK and PD properties of a proposed biosimilar and the reference product. So the developed biosimilar bioanalytical methods used for PK and PD evaluations should be accurate, precise, specific, sensitive, and reproducible. Also, the safety and immunogenicity data from the clinical pharmacology studies should be collected and evaluated. The overall immunogenicity assessment should include relevant patient populations that are not immunocompromised and thus which are able to mount an immune response. So these are all the critical considerations to be performed in clinical pharmacology studies. Next. So the first and foremost comparative clinical study is PK/PD similarity. So pharmacokinetics investigates how the drug deals with the drug in contrast to PD, which examines how the drug deals with the body. The time course of drug concentrations in serum, plasma, or any other biological fluids, obtaining information on ADME and the drug exposure of the patient. So understanding PKPD is a key element of drug development, and PK assessment is therefore included in preclinical and clinical studies. PK studies are very much important to characterize drug exposure and bioavailability to predict dosage regimens as well as changes in doses, changes in dosage regimens to describe individual subject variability and to establish safety margins. So overall, in clinical studies, PK assessment is primarily required for dose escalation and dose finding studies. The PK of the drug generally depends on patient-related functions such as individual physiological characters as well as the drug's biochemical properties as well. Next slide. So the design and development of PK assays is an important factor for conducting comparable clinical studies. The most common format of LBA-based PK assays is the sandwich principle. In principle, the analyte, like therapeutic protein or MAB, is sandwiched between the capture antibody, antigen or soluble receptor, and the detection antibody, antigen or soluble receptor. And these assays can be usually performed in three different versions, either antibody capture assay or antigen antibody capture assay or soluble receptor capture assay. The antibody capture assay can be applied for any biotherapeutic drugs, and the prerequisites to perform antibody capturing is the availability of pair of antibodies directed against the analyte. In case of antibody drugs, an anti-idiotypic antibody is usually immobilized on the plate and is used to capture the drug, while a secondary antibody is labeled with an appropriate tag and is used to detect the drug. In case of antigen capture format, especially it will be used for the maps and the immobilized antigen is used to capture the drug onto the assay plate, which is then detected using a species-specific anti-FC antibody labeled with an appropriate tag. So many therapeutic proteins in drug development binds to receptors with high affinity that either circulate in blood or an integral part of cellular membranes. If these receptor proteins are available in a soluble form, a soluble receptor capture assay is more preferable assay format. So in PK and toxicology studies, it is often essential to estimate the different forms of the drug which are present in biological fluids, whether it is a free or partially free, bound, and total drug. So the level of the free drug reflects the availability of the active drug, while the levels of the bound and the total drug are mainly used by toxicologists to estimate the safety of the drug. So what is free form? So therapeutic antibody that has both antigen binding sites free is considered as a free drug, and the antibody that has only one antigen binding site will be considered as partially free drug. And the bound form is the therapeutic antibody that is occupied with both the antigen binding sites. And total is a sum of free, partially free, and bound drug. And the choice of assay depends on the requirements by considering the combination of different anti-idiotypic antibodies by utilizing competitive versus non-competitive idiotypic antibodies. Competitive anti-idiotypic antibodies can only bind to the antibody drug in the absence of the antigen, whereas non-competitive anti-idiotypic antibodies bind to the antibody drug independently of the antigen. Hence, the selection of the appropriate combination of antibodies is crucial in estimating the required forms of the drug. Next slide. So a PK method can be described as a procedure for the quantitation of drug concentrations in a biological matrix over time after dosing in a clinical or non-clinical study. So the bioanalytical PK methods for establishing biosimilar comparability or bioequivalence in clinical studies for regulatory submissions are often developed to be drug specific and are always validated for their intended use. But the question is whether to develop a single assay or two assays for estimating biosimilar and reference drug concentrations in biological samples for demonstrating biosimilarity. So the main differences between one assay and two assay approaches are listed here. Usually, one assay approach is a conservative approach to use biosimilar curve and estimate both biosimilar and reference drugs in the respective samples. Whereas in two assay approach, concentrations will be calculated from curve of the same respective materials, and then we have to analyze the blended samples using two calibration curves. In case of one assay approach, the assay variability will be minimal and or it will be zero. In case of two assay approaches, because two assays possess different properties and the variability between the two assays is high. Blended study sample analysis is possible in case of one assay approach, whereas in two assay approach, blended analysis would require all samples to be run through both the assays, which is time consuming and cumbersome in nature. For one essay approach, it's the method development is easy and it's very easy to validate. Whereas in case of two essay approach, we have to develop two methods and perform two validations, which is a time-consuming process. So overall, it's always recommended to utilize one essay approach for establishing PK similarity for the biosimilar drug molecules. A well-developed, robust method should be capable of quantifying both the biosimilar and the reference compound comparably within estimated SL limits. The use of a different PK assay between the reference and biosimilar could add more complexity to bioanalysis planning and to data interpretation. Therefore, the recommendation is to use one PK assay for comparison of both reference and biosimilars. Next slide. So the assay data obtained during development should be interpreted carefully before moving into the validation phase. So the following key aspects of method development are expected to influence the outcome of the PK assay used to determine bioanalytical similarity. So during development phase, so many parameters needs to be taken into considerations for developing a robust assay. When it comes to essay methodology or platform, it's very important to choose an appropriate essay platform. The inherent nature and properties of the molecule under evaluation should be considered first. Additional parameters such as presence of endogenous versions of the drug, circulating antidrug antibodies, assay sensitivity, required dynamic range, and available sample volume should be taken into account. So another aspect is selection of the critical reagents is very critical. Usually these are antibodies, receptors, target proteins, and ligands that are specific to the drug in the assay. The nature and stability of the critical reagents used in the assay should be studied in detail during development, and well-characterized reagents should be used in validation and sample analysis. These reagents may be generated by using either a biosimilar drug or a reference, depending upon the immediate availability of the drug and evaluate the specificity. While selecting the calibrators and QCs, a standard calibration curve is prepared by spiking known concentrations of the biosimilar or the reference therapeutic in the relevant biological matrix. This curve should consist of at least 6 calibration points that should cover the entire estimated drug concentration range. The standard curve calibration point should be placed approximately equidistant on the logarithmic scale from each other. Also, if required, anchor points can also be utilized, which are below and above the limits of quantitation to improve curve fitting in development phase and can carry it into the validation and routine sample analysis as well. Quality control samples are prepared by tracking known concentrations of biosimilar and reference in the related biological matrices. It is recommended to evaluate QC samples in at least 5 levels, including low, mid, high, LLOQ, and ULOQ concentrations of both drugs separately. Also, dilution QC should be evaluated as per the requirements of the study. So it's very important during the development phase of LBA that the standard curve generated using biosimilar drug should be bioanalytically similar to the standard curve generated using reference drug. So the recommendation is to use an assay design that minimizes potential experimental bias in positional, operational, and environmental effects. The calibration point should be analyzed with the same set of assay reagents in the same biological matrix under the same assay conditions in at least three independent runs made by multiple operators over multiple days. So at least three independent calibration curve preparations, each of the biosimilar and reference should be analyzed in duplicate on the same assay plate. So once the calibration curve similarity was proved in the development phase, a systemic evaluation of QC results using single calibration curve from the biosimilar is recommended during the validation phase. So the nominal concentration of QC samples generated using biosimilar drug should be bioanalytically similar to the nominal concentration of QC samples generated using reference drug. So it is recommended that interbatch and samples and the intrabatch accuracy and precision assessment should include at least six independent runs made by two or more operators running over two plates over 3 days in a balanced fashion to provide a high degree of statistical assurance and detect potential bias due to unforeseen regions on a particular day or operator. So as per the plate layout, you have to use the standard code from the biosimilar direct and utilize QCs from both biosimilar and the reference drug with multiple replicates to see whether it is meeting the acceptance criteria of percent RE and percent CV is should be within plus or minus 20% of nominal for high, mid, and low QCs, within 25% for ULOQ and LLOQ QCs. Also, the total error should be less than or equal to 30 for the nominals of high, mid, and low, and less than or equal to 40% for the ULOQ and LLOQ QCs. So once the method is developed and validated and able to achieve the targeted acceptance criteria, then the same method will be utilized for conducting study sample analysis to estimate clinical endpoints. So usually the primary clinical endpoints will be the Cmax, AUC 0 to infinity, and secondary endpoints like AUC 0 to time and Tmax, T off, and all the clearance parameters. So this primary endpoint should be considered for both PK and PD similarity. Also, the average equivalence approach should be considered, which involves a calculation of 90% confidence interval for the ratio between the geometric means of the parameters for proposed biosimilar and the reference product falls within 80 to 125%. So this is the criteria usually followed for small molecule BA/PE applications shall also be applicable for biosimilar PK similarities. Next slide. So the next important assay is the immunogenicity assays for the development of biosimilars. So immunogenicity is a humoral cell-mediated immune response to any biological product. The majority of biological and biotechnology-derived proteins induce an immune response that may be triggered by patient, disease, or product-related factors. So the clinical consequences of an immune response to biotherapeutics may range from no effect to benign reactions or loss of efficacy and even serious life-threatening events. Therefore, testing of unwanted or undesirable immunogenicity is critical for any biotherapeutic drug development program, including those for biosimilars. The immunogenicity evaluation in the clinical setting is conducted using anti-drug antibody assays to detect the presence of antibodies that bind to the biotherapeutic product in a biological matrix, and neutralizing antibody assays which demonstrate whether the binding ADAs neutralize the biological activity of the biotherapeutics. So the impact of the formation of ADAs will have an impact on other parameters like alteration of PK, either for sustaining or clearing the drug in a faster manner, or it will also have the impact on PD by loss of efficacy due to the neutralizing activity or endogenous cross reactivity, and also the treatment outcome, whether the patients are patients or subjects or responders or non-responders, and it will have sometimes safety aspects also to show infusion reactions or hypersensitivity or anaphylaxis reactions. So usually immunogenicity testing will follow a tire-based approach utilizing three tiers starting from screening assay. If the subject samples are positive in screening assay, it will go for a confirmatory assay. And then if we get the positive samples in confirmatory assay, it will undergo tier 3 approach for titration and then for further characterization for NAB or domain specificity and isotyping. Next slide. So one bioanalytical challenge of biosimilar drug development is how to demonstrate that the anti-drug antibody assay can adequately detect antibodies against both biosimilar and originator in the one assay approach, and to demonstrate comparable rates of immunogenicity between the originator and the biosimilar. The regulatory guidance on the practical conduct of immunogenicity studies in the biosimilar context is very limited, and so far there is no consensus in the bioanalytical industry. The use of one assay or two assays for immunogenicity assessments has been widely debated in the bioanalytical community for the past few years. Each approach has its own advantages and disadvantages, but what is critical to demonstrate in order to have full confidence in applying a one assay approach using the biosimilar, and how best to demonstrate the suitability of one assay approach. So the most conservative approach described in the usage of anti-biosimilar positive controls throughout the study from assay development until sample analysis. So the starting points of the outlined approach are the required reagents, whether the biosimilar originator drugs labeled biosimilar as the core reagent and anti-biosimilar antibody as a positive control. The recommendation is to know the actual drug concentration of the originator that can be found on the certificate of analysis if available, or the originator should be analyzed using the same method that is being used for the biosimilar. Otherwise, if there are any concentration differences between biosimilar and the reference product, the binding affinities of the anti-drug antibodies will also differ and it may have impact on the assay. So in the initial method development, we have to perform some basic 1SA feasibility testing by performing titration inhibitory comparabilities. And once we show that both the drugs are behaving similar with respect to binding to the respective anti-drug antibodies, then the method must be validated as per the FDA guidelines for cut point evaluation, sensitivity, matrix interference, and drug tolerance, precision, and finally the stability experiments. Next slide, please. So during assay development, a basic one assay feasibility testing should be performed, and a minimum of two separate experiments performed once good estimates of PC concentration levels, MRD, and confirmatory concentrations are available. So the basic first experiment is to perform titration comparability by utilizing low PC and high PC signal inhibition curves obtained by titrating increasing concentrations of biosimilar and originator drugs. So the suggested plate layout can be seen here by utilizing biosimilars on the reference product both from US and EMA. So in order to evaluate the outcome of this experiment, the obtained curve should be compared using visual or statistical assessments. The aim is to demonstrate that the binding profiles of the PC towards the biosimilar and the originator are same. And also, the generated data can also be used for an early determination of the assay drug totals. The second experiment is the inhibition comparability experiment, which consists of titration of the PC at estimated confirmatory assay quenching concentrations of the biosimilar and the originator. If not yet determined, the quenching concentration should be as close as possible to the one that will be used during the future confirmatory assay. And the suggested plate layout is also represented here by utilizing biosimilar and the reference products. So the aim of this experiment is to demonstrate that the assay is able to detect ADS across a given antibody concentration range in study representative confirmatory assay steps. So in the In the graphs, the originator and biosimilar are very comparable in nature, and it is proven that ADA binding comparability is same between originator and biosimilar. So, in addition, it is also recommended to assess drug tolerance for both drugs and with the criteria of an acceptable two-fold or three-fold difference between the biosimilar and the originator. The development feasibility testing should generate confidence in the one assay approach and supports the totality of the bioanalytical data from development and validation to generate ADA binding comparability data. Next slide. So, coming to neutralizing antibody assays, immunogenicity assessments typically use a tire-based approach, which requires validated methods for the detection of binding as well as neutralizing ADAs. The complexities reside in the fact that some NAB assays are generally qualitative in nature and are highly susceptible to drug interference, and sometimes it's very difficult to set up in the presence of matrices, especially the cell-based assays in particular. The potential clinical outcome of immune responses may have consequences on efficacy because of the nature of the NABs will block the efficacy of drug products by targeting epitopes which are critical for efficacy. NABs may also cross-react with a non-redundant endogenous counterpart, thereby blocking the biological function of the endogenous protein. They may also cause anaphylaxis reactions, which are acute allergic reactions characterized by certain clinical features, as well as cytokine release syndromes. These are the reactions caused by the rapid release of pro-inflammatory cytokines, and also non-acute reactions like delayed hypersensitivity and immune responses, which are secondary to immune complex formation. So the obtained outcomes may potentially lead to termination of a biological therapeutic drug development program. Hence, in the context of biosimilar, it's very important to establish comparability. It is necessary to determine the incidence of NAB to the originator and biosimilar. And it's very critical to link the immunogenicity to the clinical results, whether there is an impact on PK results and associated adverse events. So, depending on the risk associated with the type of drug and its mechanism of action, the choice of the format of NAB assay should be assessed. The NAB assay for selection should be scientifically driven approach that relies on combination of three factors, mainly the mechanism of action, evidence of desirable assay performance characteristics, and risk of immunogenicity. Next slide. So there are two formats currently available for performing NAB assays. Which one to choose? Cell-based versus non-cell-based competitive ligand binding. So cell-based assays provide a functional readout using relevant cell lines, which is in close reaction to the biological function of the drug, and thus may more closely reflect the in vivo situation. However, cell-based NAB assays are difficult to establish as suitably relevant cell lines a proper assay be verified. An additional challenge with cell-based assay is assay sensitivity and consistency across programs. This is the most critical part as the cellular response has to be robust and specific. In addition, the cellular response may be impaired by many factors such as matrix interference and the presence of drug product. Non-cell-based or competitive ligand binding assays are relatively easier to set up with higher assay precision and better sensitivity, as well as having the option to use different detection systems. Therefore, if the nature drug under our physiological conditions results in certain roadblocks, then a non-cell-based assay must be developed. So, although EMA and FDA immunogenicity guidelines recommends the use of cell-based assays for the detection of NABs, non-cell-based assays are considered more appropriate when the drug's mechanism of action is to block a soluble target or soluble receptor. Next slide. So the bioanalytical industry recognizes that it's very challenging to develop and validate immunogenicity assays to support comparability studies for biosimilar drug product. In spite of having advantages and disadvantages in using one assay approach or two assay approach, The recommendation is to use one assay approach for biosimilar NAB detection. So if one assay approach is used, it is recommended to use the biosimilar set of reagents to conduct assay development work for determining cut point, sensitivity, selectivity, and precision. So the evaluation of key assay attributes must be conducted during assay development stage to ensure the assay can detect the NAbs against both the biosimilar and the reference molecules. It is recommended to use one representative batch of the biosimilar or the originator for assay development, validation, and even sample assessment studies. Depending on the NAB assay strategy, specific requirements need to be conducted to demonstrate that there is bioanalytical equivalence of the biosimilar and the originator products. So equivalence is nothing but bioanalytical comparability assessment, which should have a statistical basis for the final conclusion. If one assay is selected, it is important to show that both biosimilar and the reference give comparable qualification results within the assay. And if two assays are selected, then it is important to show that both assays have similar sensitivity and specificity, which can yield comparable results. NAB assays are based on the ability of ADA to neutralize drug's biological function. Therefore, it is critical to find an optimal drug concentration in the assay system, which allows for a sufficient bioactivity signal, and the concentration selected allows optimum neutralizing effect by the APC. Both biosimilar and originator drugs should be used in experiments to ensure that they are comparable in the assay. And MRD evaluation is also one of the critical parameters and should be assessed as a part of initial assay development experiments. MRD must be carefully selected to ensure that the integrity of the matrix is conserved as well as to demonstrate that the matrix does not interfere in the assay. So in the case of cell-based assays, it is especially helpful to evaluate the optimum MRD against a buffer curve to ensure no matrix interference exists. In order to achieve this balance, multiple concentrations of matrix should be tested in the assay. The sensitivity of the assay must be reported after factoring the MRD or matrix dilution factor. Sensitivity results should be compared to demonstrate that the drugs behave similarly in the assay. Also, drug dose response curve should be tested using serially diluted drugs, utilizing biosimilar and the reference to check for a similarity and to prove there is a consistent range of response across two drugs. The drug concentration, which gives a strong enough signal over background, usually 2 to 5 fold over no drug baseline, but it should not be in the plateau region of the response curve. Accordingly, it should be chosen for further assay development work. Usually, the sensitivity of a typical NAB assay of at least 0.5 microgram per ml should be acceptable and recommended as per the regulatory guidelines. It is important to evaluate drug tolerance for both biosimilar and originator compounds in the NAB assay. However, it should be noted that due to non-qualitative nature of NAB assays, the difference of 2 or 3 fold may be acceptable. So once the method is developed, it should be appropriately validated for determining assay cut point, sensitivity, selectivity, drug tolerance, assay precision, benchtop, and even freeze-thaw stabilities. All these parameters must be assessed in validation phase. The assessment should be made as per an approved assay validation plan or protocol with a defined target assay acceptance criteria for each assessment. So there are no specific guidance from the regulators or white papers on how to conduct confirmatory and NAP titrating assays, especially in biosimilar programs. However, if samples generate an unexpected or non-specific assay response, the reactive samples need to undergo further testing, including confirmatory assay or titration. Usually, for drugs that are categorized as high-risk agonists, such as cytokines or growth factors, the reporting of titers would be important to evaluate the potential for clinical risk to patients due to neutralization of the endogenous chemical. If that is done, the NAB data should be reported by performing twofold, threefold, or any higher serial dilution of the sample till the NAB response signal is below the cut point. So depending on different methods and their respective capabilities, the titer can be reported as a reciprocal value of the highest dilution of the sample that results in a signal above the cut point value. So overall, for majority of the proposed biosimilar products in the regulated markets, presenting a solid bioanalytical package is critical in biosimilar drug development in order to support PK, PD, immunogenicity, and safety or efficacy trials. Next slide. So coming to nutshell, the critical takeaways or conclusions from this webinar would be a systematic process for assessing the degree of bioanalytical similarity between biosimilar and reference by developing robust bioanalytical methods. The two-stage process consists of a development phase and a validation phase with targeted acceptance criteria must be provided for evaluation of analytical similarity. Also, the recommendation is to use one assay approach for demonstrating biological similarity in terms of PKPD, immunogenicity, and NAB assays so that we can save some time and money as well. Clinical pharmacology studies are part of a stepwise process for demonstrating biosimilarity between a proposed biosimilar product and the reference product to show that there are no clinically meaningful differences between the products. So the regulators require the minimum data sets of analytical similarity, non-clinical toxicology, human PKPD similarity, comparative clinical efficacy, and safety studies in generating totality of evidence to demonstrate biosimilarity for the marketing approval of biosimilars. Next slide. So these are some of the regulatory guidance documents which needs to be referred for the complete set of developing biosimilars for the regulated markets. Next one. Yeah, that's all from my side. Thank you all for your active participation throughout this session.
Thank you, Dr. Jagdish. That was very insightful. Now, moving on to the next session, we would like to answer a few questions. We have some wonderful questions presented here. Our first question is, what are the key challenges in developing and validating immunogenicity assays for some of the newer drug modalities? Dr.
Yeah,
Jagdish, can you
so
please answer that?
newer drugs may elicit more immune response that demands additional immunogenicity assessment strategies. For example, in case of ADCs and bispecific antibodies or fusion proteins, in addition to the screening, confirmation, and titration assays, they may require domain analysis and isotyping to understand the source of immune responses. Often, it may require two or more independent neutralizing antibody assays, which is more challenging to develop bioanalytical assays. A more scientifically driven and risk-based approach is recommended for developing such assays.
Thank you, Dr. Jagdish. Our second question would be, what are the possible disadvantages of using one assay approach for NAB analysis?
Yeah, that's a great question. The major disadvantage of one assay approach is that due to the potential differences between the biosimilar and originator drug molecules, for example, different glycosylation or other post-translational modifications, the ability to detect NAB against unique structure of the originator may not be detected if only one set of reagents, that is the reagents of the biosimilar drug, is used. So one important explanation to this point is that since the regulatory agencies only require that the biosimilar product is no more immunogenic than its originator product, Using biosimilar reagents in one assay approach will be the most conservative approach.
Okay. A third question for the evening is, if an assay is suspected of endogenous molecule interference, what are the recommended steps to address this issue?
Uh, it's initially recommended to verify whether the interference is from endogenous molecule or general matrix components. Endogenous compound interference is often noted in studies involving replacement therapeutics. To address this, options include usually commercially available endogenous free matrix or depleting endogenous compounds in the matrix to create an endogenous free matrix. Alternatively, obstructing endogenous background signals is another effective strategy for eliminating endogenous interference.
Thank you. Uh, can we please have the thank you slide back on? Okay, so at this moment, I would like to thank everyone, my panelists and my audiences for the questions. At this moment, we won't be able to take any other questions. I understand this session has kept us engaged, but there are certain time constraints. So, but don't worry, we'll be sending you all the questions through the emails. And till then, lastly, but on the behalf of Veeda Group, I would like to sincerely thank everyone for being here and taking out this time to attend this webinar. Especially, I would like to thank Dr. Jagdish for being a wonderful presenter and giving us all great learnings. We at Veeda Group earnestly look forward to host you again. Till then, thank you and take care.